Welcome, everyone. I'm Amanda Egan, the Director of Patient Engagement and Clinical Programs with the RUNX1 Research Program. And I'm here to kick off the webinar, Introduction to the RUNX1-FPD imatinib Clinical Study. We're so glad you're all here. We're also really thrilled that the very first clinical intervention trial for RUNX1-FPD has launched, and that we have the two study leads here today to speak about the trial, as well as two other of their team members. They'll discuss the rationale, who's eligible, and a bit about what a patient can expect when participating. So Dr. Lea Cunningham of the NCI is the principal investigator, and Dr. Alan Cantor of Boston Children's is the associate investigator. And many of our patient community know Dr. Cunningham well from her history with the RUNX1 Natural History Study at the NIH. And Dr. Cantor has been a friend of RRP for many years. We've supported his work since 2017, and some of his work has laid the foundation for this trial, and he'll get into that in a bit. And then joining at the end of Dr. Cunningham and Dr. Cantor's presentation will be a couple of people from the study team to help answer questions. That will include Rebecca Alexander. She's the research nurse for this study. Her role is similar to Katie Crafts with the Natural History Study. You may be familiar with Katie. And Yi Liu is the genetic counselor working on this study as well, and her role is similar to Natalie's on the NIH Natural History Study. Please note that Dr. Cantor and Dr. Cunningham prepared their presentations specifically for a patient audience, as we hoped they would. We know that there are some clinicians and scientists who registered for the event today. We'll do our very best to answer their questions as well, but the primary goal today will be to ensure patient questions are answered first. So I'm just going to cover a couple of housekeeping items quickly. So during the presentation, please use the Q&A icon as you see here to type in your questions. The questions, therefore, will only be visible to me and the speakers at that point. Feel free to use the @ symbol to direct your question to any specific member of the team. And in that Q&A icon window, you'll be asked, you have the opportunity to indicate whether you want your question asked anonymously. We're just thinking about patient confidentiality and privacy here. Regardless, I'll read your question aloud without mentioning your names. And then we encourage you to ask any question you have, no matter how simple, because, as if you're wondering it, others are likely wondering it as well. And then finally, please recognize that Dr. Cantor and Dr. Cunningham's and Rebecca and Yi's responses should not be considered personal medical advice, of course. And with that, I will let you all take it from here. When you're finished, I'll return to facilitate answering questions.
Well, thank you, Amanda. Welcome. Hi, I'm Alan Cantor from Boston Children's Hospital, and thank you for joining today. As Amanda said, I'm going to begin by just going over some of the rationale and background for the trial, and then I'm going to hand it over to Lea, who'll really go through some of the nuts and bolts of the trial. So as probably many people know here, that we all get 2 copies of every gene, or essentially every gene from our parents, one from your mother, one from your father. And in RUNX1-FPD, one of those 2 copies of the RUNX1 gene is defective. So that's shown kind of schematically here at the bottom. The RUNX1 gene is on chromosome 21. And so one of those 2 copies isn't working. And so you have an overall deficiency in the amount of RUNX that's in your cells. But importantly, there's one normal copy that remains. And so one of our approaches is to see if we can find drugs to enhance the activity of the residual normal RUNX1 protein. In other words, try to juice up this the normal RUNX1 to try to compensate, or at least partially compensate, for this deficiency, and thereby to reduce the risk of developing leukemia. And so again, since this gets really to the heart of the problem of RUNX1 deficiency, this should also help with all the other aspects of the disorder, including the platelet disorder and the inflammation. So I'm just going to give just a little background about RUNX1 itself. So as probably a lot of you know here now, that RUNX1 is called a transcription factor. And what a transcription factor is, it's a protein that binds to DNA in different places in your genome and on your chromosomes, and it turns on and off genes nearby where it sits. And like many proteins, RUNX1 has different parts of it that do different things. And so first is this region in red called the RUNX domain, and this is a part of the protein that binds to DNA. There's this blue region here called the transcriptional activation domain. This is important for turning on the genes. And then there's this region here, the green region called ID here, which I'm going to really focus on, that really regulates RUNX1 activity. So a number of years ago, my group and others discovered that some proteins called kinases can chemically modify RUNX1 in this regulatory region by chemically adding a phosphate group or several phosphate groups to this area. And when that happens, this puts RUNX1 in sort of an inactive or off state. But there are also other proteins in the cell called phosphatases that can chemically cleave off that phosphate group. And then this relatively turns RUNX1 back on. And that normally is really kind of a balance going on between these phosphates being put on by these kinases and removed by the phosphatases. This probably reflects different signals that the cell is getting at any given time. But importantly, in work that was supported and funded by the RUNX1 research program, my group had discovered that a drug called imatinib, also known as Gleevec, can inhibit this kinase that adds that phosphate group to the RUNX1 and normally turns it off. And so if we can inhibit this kinase with imatinib, we'll have less phosphates getting put on, and these phosphatases will remove it and then relatively activate RUNX1. And so one way to think about it is there's normally like a balance of RUNX1 being inactive and active at any given time, but by treating with imatinib, we're going to tip the balance towards a more active state and thereby kind of juice up the available RUNX1. Or another way to think about it, we're kind of going to sort of dial up RUNX1 activity, and we probably aren't going to get you completely back to 2 copies of the gene, but we think we can get to about like 1.5 copies. And this is, we think, is probably going to be good enough to hopefully have some important effects. And so I'm going to show you just 1 or 2 pieces of data just to sort of illustrate this. So what I'm showing here, this is a graph that we're looking at RUNX1 activity on the y-axis. And I just want you to focus on the blue for right now. And so we've added increasing amounts or concentrations of imatinib into this test tube and measure this RUNX activity. And you can see as we increase this imatinib, we can boost the activity of RUNX1 up here. This green line, the control, is important because we want to make sure that this isn't happening through something that doesn't require RUNX1, and that's shown here. So all the effects we're seeing is shown in blue. It's really happening through RUNX1. And overall, again, we can get about a 50% boost of the RUNX activity with imatinib. Of course, it's important to then test this in what we call preclinical studies. So before going into people and humans, we want to test this in various cells in the culture or animal models. And what we really want to see now is, can imatinib rescue some of the defects associated with RUNX1-FPD? And we've done this a number of ways, and I'm just going to show you just one for the purposes of today's talk. And this is work we did in collaboration with Anna-Priya Agarwal at Oregon Science and Health University and Paul Liu through the NIH Natural History Study. So what we've done is we've taken bone marrow cells from patients with RUNX1-FPD collected through the Natural History Study. And you can take those cells and you can grow them in the Petri dish into megakaryocytes, again, because the bone marrow contains the precursors for this. And we know that with RUNX1-FPD, we don't get as many mature megakaryocytes. And those are the cells that produce the platelets, and that's why we have low platelets and the platelets don't function well. So we can take these cells, and now when we grow them, we can either do it in the absence or the presence of imatinib and see if we can rescue that megakaryocyte maturation, so try to improve this. And the results of that study are shown on the right. So just first on the left is an untreated cell, and what we're looking at is the percentage of what we call mature megakaryocytes in the culture. And I apologize, this was actually a little bit backwards, but if I'm going from right to left, we added increasing amounts of this imatinib, and you can see that this led to more mature megakaryocytes, so we were rescuing some of the defects. So again, we had other data looking at RUNX1 deficiency in other ways, and they all sort of gave some similar types of results. So just a little bit about imatinib or Gleevec. So imatinib was first FDA approved in 2001. In fact, it made the front page of Time magazine then, because this was really the first example of what's now called precision medicine, which knowing the exact target and designing a drug is really having a major impact. Imatinib is now currently FDA approved for treatment of at least 6 different types of leukemia and actually some other disorders as well. It has relatively low toxicity, and Lea will talk a little more about that. And again, we have a very long 20+ year experience of this drug in patients, and many of these patients take it very long term. These are patients with chronic myelogenous leukemia. And importantly, we know that the bloodstream concentrations of imatinib in patients who are taking a standard dose, which is 400 milligrams once a day, correspond to the levels that we've shown seem to improve RUNX1 activity in our experiments. So in other words, we should be able to achieve that increased activity in humans. And then one last point is the Gleevec patent expired in 2016, and it's now available in generic. And this is important because it actually reduces the costs of this kind of a treatment. So with all this in mind, we've developed a phase 1b clinical trial of imatinib. And again, the idea is to try to enhance the activity of the residual normal copy to compensate for this loss and ultimately reduce the risk of leukemia development and as well as the other problems associated with RUNX1-FPD. And we're very excited that this trial is opened the end of last year, and we're now enrolling patients. And with that, I'm going to stop and I'm going to hand it over to Lea, who's going to tell you more about the actual trial.
Thank you so much to RRP for the invitation to join today, to all the participants who have joined us today, and to Alan Cantor for the excellent foundation and background. So I believe I'm now sharing my slides. If someone could just let me know if they see them when I put them into presenter mode, that would be great.
Can see them, I just don't see presenter mode yet.
Okay, no problem. I was afraid if I hit presenter mode, I might obscure my view of the participants for a second. So I'll go ahead and do that now. Are you still seeing the correct view?
I think you want to reverse it because we can see your.
All right, how's that?
Oh, now we just see you. I think you duplicated. Let's see.
Okay, best laid plans. Let me try this again.
It always works perfectly until you have your video on, right?
Yeah.
Okay, there it is.
It looks okay now?
Perfect.
All right, great.
Yep.
All right, thank you so much. Sorry about that. Okay. Okay, great. So basically, I just wanted to step back a little bit and think about considerations of joining a clinical trial in general. And so for many folks on the call who may already be familiar with the natural history protocol or enrolled on it, these are things that they have probably thought about before, such as, is the research of interest to them? What are the perceived benefits and risks? What are the logistical considerations? And honestly, this is probably the one that we get the most in terms of time and energy required to participate. This study is a little bit different because it has 2 study consents. One is for screening to make sure that folks would be eligible to participate. And then there's a separate consent for actually receiving imatinib. And then in general, any questions or concerns. And taking another step back to think about trial design in general, as you heard from Alan Cantor already, imatinib has been FDA approved since approximately 2001 for several different illnesses. And so some of those illnesses include things that folks with RUNX1 mutations would already be treated for. So under those circumstances, if somebody had a germline RUNX1 mutation and they also had a chronic leukemia, then they would be eligible to receive imatinib, no problem. And so what would occur there is prior to 2001, imatinib was evaluated very carefully under all of those different study populations, everything from preclinical modeling through phase 1 and 2 and 3. The approval would have come here in 2001. And then there's a significant amount of phase 4 studies or post-marketing studies that go on to evaluate for any side effects that are noted when the patient population increases significantly. So you can see, these are usually preclinical studies here, like in under a microscope, cells from bone marrow or cells from a skin biopsy, for example, and then slowly moving up to a very small amount of participants to make sure that things look safe and tolerated, and increasing the number of folks who are included as the phases go up. Now, of course, for a rare disease, we're not ever going to get up to these very, very high numbers. And so the designs can be different for those types of trials. But the reason why we're doing this trial in folks with germline RUNX1 specifically is because the imatinib has not been studied at lower doses for this particular patient population so far. So the purpose of the study is to identify whether it's safe and also tolerated at these lower doses. And most importantly, we want to know if it's helpful and starting to build any information about whether bleeding symptoms change, whether clinical laboratory studies change. So those could include anything from platelet counts to how well the platelets function, like with aggregation or electron microscopy to look at granules or other clinical laboratory studies like that. We are hoping to look at imatinib blood concentration levels now in the second half of the study to make sure that the concentrations that we're seeing in the blood, how they're similar or different to previous studies for previous populations. And then also there are research studies to figure out if RUNX1 activity changes before and after imatinib. So all of these studies help us learn how imatinib works for folks who have RUNX1 mutations at lower doses than what have been studied previously. So the criteria, since it's a phase 1 and a safety tolerability study, is to start with adults to make sure that folks have platelet counts that are 65,000 or higher, and for the most part, no major medical problems like cardiovascular disease, heart disease, kidney problems, liver problems, pregnancies. And so we're hoping that folks would have either a pathogenic or a likely pathogenic mutation. So at this point, we're not accepting folks who have a variant of unknown significance, and that folks who are participating would have the type of germline RUNX1 variant, such as a deletion or something that is causing a haploinsufficiency versus a dominant negative. And so just to kind of make the distinction, to go back to Alan Cantor's dial, that a haploinsufficiency would be more like having one copy, but not one copy that's also trying to interfere with the normal copy, which is what you would see with the dominant negative effect. We're hoping to enroll participants who have either a history of bleeding or an abnormal laboratory study related to platelet number or function, that they are at their baseline for their bone marrow, and that their secondary somatic mutation panel. So just as a reminder, the somatic mutations are those that people acquire as they age, that none of those are problematic. So if there are benign or variants of unknown significance under this category that are less than 5%, then that would be okay. And then we need folks to be able to swallow pills, since imatinib comes in a pill form. And in terms of the timeline, we're noticing that there are a lot of questions. And so reaching out early and often about the trial so that questions can be answered is probably the best way to go forward. And again, there are both a screening consent form in addition to an administrative consent form. And so from a practical standpoint, we've noticed that folks who may already be at NIH for their natural history study may wish to consider hearing more about the imatinib trial and the screening study while they're here. And the reason why is because that way we can use all of the same laboratory studies that have already been drawn from the natural history protocol for the imatinib screening study. So when folks get their complete blood count, for example, for the natural history protocol, we can use those results. We don't have to poke anybody again or get any additional blood. There are just a few little things that we add on. So we add on the TSO 500, which is the sequencing panel for 500 other genes. And then we add on a few infectious disease serologies, such as hepatitis, which is a liver infection. And then we also add on kind of a sticker test to the heart to look for heart rhythm, also known as electrocardiogram. For anyone who wishes to return to NIH after those results come back to consider starting imatinib, then they're welcome to do so. And we're trying to make this as easy as possible, depending on what people wish to contribute. So, for example, for folks who would be coming in for the first half of the study, then they could fly in and out the same day, for example. They don't even have to stay overnight if they don't want to. We have noticed along the natural history protocol that January, February tend to be, times of the year that people don't want to come to the mid-Atlantic, Maryland area. But once the weather starts getting a little bit nicer, the cherry trees are in bloom, the Smithsonian, museums and zoos and all of that are here, then, sometimes people find it, better to visit at those times of year. And for people who wish to donate marrow, which is optional on the study, they could consider staying an extra day, for example. So this slide is a little bit busy. I apologize. So just try to bear with me for this one. So we're going to start here kind of at the purple star. And what I was seeing before about the dose for this new population is really the focus of the study. So basically, as Alan Cantor said earlier, 400 to 600 milligrams is the approved dose if you have something like a chronic leukemia. But for folks with germline RUNX1 mutations, we wanted to start much lower than that. We started at 25% or 1/4 of that dose at 100 milligrams, starting with 3 people at a time, and each dot is 1 person. So, for example, if we start at 100 milligrams for 1 month for 3 people, and there are no side effects, then we watch them for 28 days. And if that all goes okay, then those folks are basically done. And then we have 3 new people receiving 200 milligrams. And if that goes okay, we watch them for 28 days. And then here I threw in, a special scenario. So under these circumstances, we are getting 300 milligrams. And 3 people, but the last person had a side effect. So, okay, gosh, is that related to imatinib? Is it not? What's the best way to evaluate that? So, in classic 3+3 design, 3 additional people are enrolled, and they also received 300 milligrams. And in this case scenario, it ended up not being related to imatinib, in which case we were able to go ahead for 400 milligrams for 3 people. And at this point, the first of the studies, so this hormone dose escalation has identified, in this example, the 400 milligrams is the correct tolerated dose. And then we confirm that in the second half of the study, which is the arm 2 expansion cohort, to make sure that what we think is the right dose is for 12 additional patients for 12 weeks. So in terms of imatinib potential side effects, all of the published data is for the higher doses that were given to the folks who have chronic leukemias, for example, at 600. And so we can only guess that some of the side effects might be the same things that we might see at lower doses, like the 1 and 200 milligram dose. And using more of these up to 400. So on clinical trials, for significant amounts of time, everything is recorded. And then it's up to the investigators to figure out if side effects are related or not, and what we do about them. So, for example, if there is gastrointestinal or stomach upset with constipation or diarrhea, or throwing up, for example, then there are medications that can be given to make those symptoms better. So like Zofran is a medicine, or ondansetron is a medicine that can be given for that. So everything right here in blue is in response to a potential side effect that can be evaluated. So hydration and electrolytes. If imatinib makes people feel sleepy, and they take it at night, if it makes them have trouble sleeping, they can take it in the morning. And so the same thing is true of some methicone and Tylenol and lotion. And if a medication just is not tolerated, then in addition to very careful evaluation and talking on the phone and telehealth visits, stopping the medication is absolutely the thing to do. Our goal here is to learn if imatinib is tolerated, and there are side effects of the medication. It's not, and that means that it's not the right dose for us. So all of these things will help us learn. And all medicines do have side effects, including over-the-counter ones as well. So it's very important to be careful and to monitor people closely, and to have good back and forth communication. So frequently.
Can you hear me? Sorry, this is Amanda. There has been a little bit of breaking up of your voice periodically, and so I thought I'd interrupt you at this point to see if you have an option to call in. Sorry to pause your flow.
Yeah, no, absolutely. I will call in, and then maybe Alan Cantor wants to go through these slides, so we're not wasting any time. Starting here.
Alan, are you available? Thank you. Okay. Great.
Yep. Yeah, let me jump ahead.
Sure, I can stop sharing too, so you can pull them back up if you want. Is that better?
I think I got him, yeah.
Okay.
Okay, great. Yeah, so again, these are just sort of some frequent questions, and the way we sort of designed this. So we try to minimize how much time is needed at the NH as much as possible, as Lea already discussed. Really, it's that first day there, and we are hoping people would come back at the end of the 28 days to have more blood drawn, to look at these changes in the these markers. Most of the study visits are going to be via telehealth from home, by phone, or something called LabCorp. We will give slips to get the lab work done locally. That insurance and copays will not be required for any of this. That clinical care, consults, and close monitoring will be provided during, of course, throughout the trial and while you're taking the medication. The NIH assists participants with travel, lodging, and meals. And then really, the goal of this study is really to hope by participating, we'll really identify the best design for a follow-up study, a phase 2 study, as Lea had described the general process for moving drugs forward for a disease. So with that, we just wanted to close, and we wanted to list all the important study contacts, and some of them, I think Rebecca and Yi are on the line, as we mentioned earlier. and they can help answer some of the questions as we get into the Q&A. And then you should always feel free to reach out to Lea if you have questions about the trial itself. And you can always reach out to me, and I'm always happy to talk about some of the science behind this as well. And I think with that, we're gonna, lastly, we just want to acknowledge all the people that have been involved in getting us this far. So of course, the many patients and families and referring providers, and of course the RUNX1 research program for all their support. And then all the various collaborators, many places around the country who have really contributed to all the work leading up to this and in designing this trial as well. Okay, so I'm going to stop sharing.
Thank you, Dr. Cantor, for being nimble there. And I didn't realize we only had 2 more slides left, but I think we're good. Okay, well, I think at this point, that was a really great presentation, very clear, really exciting that you guys have launched. I have a number of questions in the Q&A window now that I'll read out to you all. I think we can, Rebecca and Yi, if you'd like to come back on, you're welcome to, and you can help answer some questions, and we'll let Dr. Cunningham join when she feels she's ready. I'll mention, Dr. Cunningham, that your voice is sort of in and out, so you might be okay if you're having trouble calling in. We could just try to work with it. Okay. So first of all, there are a number of questions around benefits and risks with imatinib. So first question is there a benefit to taking imatinib at a younger age? Is it perhaps more effective at preventing cancer? Is that something that maybe Dr.
I think that's a sort of a wonderful question. I guess I have to say we don't really probably know the answer to that at this point. I mean, one thing we are looking at in these studies and hopefully future studies is, does this affect those somatic clones that come up? You know, how many clones and how they change. So it's what we'll have to see as we do the studies if it affects. But as of now, we probably don't know if there's any. You know, we don't think there is, but there potentially could be.
Okay. And I guess a follow-up question could be, would it make sense to start at a younger age, given the lower likelihood of having somatic mutations that at a higher VAF that might preclude you from participating, or, you know, is that something to think about?
Yeah, that's probably true also. Yeah. Again, this is the phase 1 study. We're trying to be very conservative and avoiding patients who have larger clones, but we would imagine in future studies, like a phase 2 study, we would include patients who have larger clones, so we can see the measure effects on that as well.
Okay, so let's see. So have those patients who have had leukemia and germline RUNX1 who have already been taking imatinib seen a decrease in bruising and bleeding? Is that something we know? I don't know that we've measured that.
I'm not aware of many patients taking that. Maybe a very small subset who have chronic myelogenous leukemia. I know Dr. Cunningham brought this up, but I'm not aware of that data.
Yeah. Okay, maybe we can. I am wondering if Dr. Cunningham is okay. I see her there, just muted. Okay, we'll give her some time to join, and then we can circle back to that if it makes sense. So probably similarly, it might be hard to answer this question. Does imatinib decrease cancer cells?
Yeah, I mean, certainly in chronic myelogenous leukemia and other leukemias, it does. You know, in those situations.
That's the goal there, of course. Okay, great. Thank you. Okay, and then this one is a little bit more about the logistics. How long after, I guess, the first bone marrow biopsy can you start the clinical trial?
I can answer this. So we would like the bone marrow biopsy to be done within 9 months.
Okay, of starting the trial.
Yes. So if someone had a bone marrow biopsy 9 months ago, they could start the study today. So just within that 9 months.
Perfect. Okay. And then I think this may have been answered after the question was posted, but is it required or do the participants need another bone marrow biopsy after the study?
We would like one. It is not mandatory, but if they are willing, then it would be very helpful for the study.
Great. Okay. And then this is another one of those questions. I think the side effects were brought up after that question was posted. I wonder if was there anything that wasn't clear in the side effects slide that we want to highlight from your perspective? I know we had some trouble with the audio. I wonder if we should pull that. Well, we could pull that back up if you all want to highlight anything there. Or if the person who asked the questions wants to follow up on any specific side effect, maybe that makes more sense. Okay, here's Dr. Cunningham. Can you hear us okay?
Hi, yes, I'm back on my phone. Sorry for the previous audio difficulties. Can you hear me okay?
No worries. No worries.
Okay, great.
It was just a little muffled here and there. So
okay, sorry about that. So I did hear the questions and the answers so far. I agree with everything that was said. I think the next question was about the side effects. We can go back to that slide unless there was a specific question.
you know what, if you don't mind, since it did there was some breaking up, maybe you could bring that back up and just highlight those again. Oh, actually, there is a follow-up question. Let's see. While you're pulling that up, there is a follow-up question about side effects. For the side effects, it mentioned many had no side effects. Do we know what % that many represented? Is that vast majority or minority?
Yeah, that is such a wonderful question. I believe that it will probably end up being dose dependent. So the published literature on the side effects of imatinib are for 400 to 600 milligrams taken daily for people who have chronic blood cancers and other indications. So I think that for the lower doses, my best guess, and this has not been studied for folks with RUNX1, is that there will be minimal side effects at the lower doses is my guess. And then one other consideration is that the reason why folks were getting the imatinib on those previous studies also had implications for their side effects. So some of the initial data with imatinib for folks who had CML, for example, there was an active question about whether or not an increased bleeding. In discussion with Brian Druckers and others who have used imatinib clinically, it was felt, and there's also some literature to suggest that it may not have been the imatinib, but rather the underlying disease, like the chronic myeloid leukemia, for example, that was more responsible for the bleeding. But the reason why we are requesting a complete blood count and looking at platelet numbers and filling out the bleeding assessment tool survey is to be as careful as possible to make sure that there are no extra bleeding and bruising or other side effects that are not tolerated. I hope I answered the question okay.
I think that was great. Thank you. Okay, we'll see if they have follow-ups, but let's see, we have a few more questions, or you know what? Oh, that's right. You did circle back to the bruising and bleeding. Thank you. So for logistics, was that one trip at the beginning, 28 days on trial, and then one trip at the end?
And it's another great question. So it depends on if folks are already enrolled on the natural history protocol. We are finding that people who are already at NIH for the natural history protocol can save some time by meeting with the study staff and having those few extra tests done while they're already here. And then, as Rebecca pointed out, they would then have 9 months to come back to start the study. And right now, the shortest amount of time would be to come in the morning and then leave, later that same day if there was not otherwise a need to be in the area for any reason. That would be the fastest that we could do it. But for those who may have heard earlier versions of this talk, like, so for example, at the RRP meeting in November, we did talk about testing the blood concentrations of imatinib in the phase 1 study. But we've eliminated that from the most recent protocol amendment so that people don't need to stay for that 25-hour period for these low doses. And we did that for 2 reasons. One, because probably the studies are not going to show us that much, number one, and number two, to make the enrollment as easy as possible on people, so to not take up too much of their time.
Thanks. While we're on logistics, I have had separate conversations with patients in the past, and some of the questions they've had around that are, can international patients participate? Is that an option?
It's a great question. The NIH can help with travel from the border. So for folks who can get to a U.S. border, then yes, but that would be something to consider, is getting to the border.
Okay, so they would just need to cover that portion of the travel. Okay, and then everything else is the same. Okay, okay, and then let's see, another question we have is, can RUNX1 patients take imatinib right now, or do we need to wait for the study results to see the benefit? Interesting question.
It's a really good question, and we've gotten that a few times. In addition to the several approved indications, there are other reasons why people take imatinib that are not "quote unquote" FDA approved. It would be theoretically possible, to do that, but then we wouldn't learn the things that we're trying to learn, like in terms of what's being changed. So, like, for example, if somebody had a prescription for imatinib, let's say they start taking 400 milligrams a day, but they're not checking their platelets or watching their bleeding bruising symptoms or their, the other study parameters, and we won't know what's different. And so the goal of doing this study is to learn what's beneficial and what's working, so that we could then be smart about designing the next study, the phase 2 study, that would be designed to "quote unquote" prevent cancer. So what does that mean? That means that we would want to monitor the variant allele frequencies, especially of the secondary somatic mutations that we know are usually problematic and involved when people have malignant transformation, for example. So that would be the overarching goal after this study.
Yeah. And also on the study, we're going to be looking at measures to see if we're really increasing RUNX activity, which will really be important to know if this is treatment is really working and it's worth pursuing.
Perfect. Yeah, that makes sense. Okay, so I love this. We have lots of good questions. Is there any concern about starting imatinib for the trial and then stopping? Is the thought that you would just re-baseline to previous levels and not regress further from current baseline?
Yeah, I think it's a great question. I'll take it too, but I have no reason to believe that you would have problems going below your baseline. I would suspect you just go back to your baseline. But Lea, I don't know if you have any other thoughts about that.
Yeah, I agree that would be what we would expect, that people would go back to their baseline. But we don't know, and that's something that that we could measure on the next trial when people are taking it longer, to see if there's a certain interval. We have talked actually about a protocol amendment to maybe, for people who wanted to come back, that we could repeat the studies and see if that's actually true or not.
Great. I have a few questions that are related to some of the eligibility. So how do you find out, or how does one find out if they have a dominant negative mutation or not?
That's one of my favorite questions. When we first started this study, I was thinking that for most of the people, especially those who have been on the natural history protocol, that might already be known. And in addition to that, at the RRP meeting in November, there are so many wonderful bench scientists who study this, and I can tell you basically that the answer is not always cut and dry. And so we have had some circumstances where somebody has a mutation where it falls into a gray area, and we're not sure while those additional studies are being run specifically for that person until we can tell them definitively. Sorry for the long answer.
That's okay. And similarly, some people are aware that they have somatic mutations, but they're not quite sure if, it's a concerning one that would disqualify them. Would you encourage them to just reach out, or what would you recommend there?
Yeah, it's a great question. For those who don't know whether or not they have secondary somatic mutations, then enrolling on the screening protocol would be a good way to find out, for example, because that's the purpose of doing it.
Okay, great. And then someone says, kind of similarly, they would assume they qualify based on the qualifications that Dr. Cunningham outlined, but they're assuming that they just follow up one-on-one for specific patient qualifications. I guess the question would be, what is the best next step if they're interested?
That's a great question. So for an eligibility question, please reach out to Rebecca or Yi or me, and our email addresses should be in the slides somewhere, and we'd be happy to help.
And we can send those out following this webinar. We can send out your contact information. I think you all agreed to share the recording, perhaps your slides as well. So all that information is there. Great. Okay. So, and someone comments, thank you for all the great responses. And then I've got a couple of interesting questions in the past. For example, one family said they're really struggling to try to figure out, where do they get involved? They're part of the NIH study, so they feel they're contributing. They don't feel like they have a great family, like a strong family history of blood cancers. Is it valuable for them to also join this study? Should they wait for another one? They're sort of trying to gauge when is I know this is kind of a hard question, trying to gauge when to take the next steps in terms of trials and intervention work. That's you.
Yeah, it's a really good question. So I guess first and foremost, thank you so much for already participating to be part of the natural history protocol. That's wonderful. I think that for anybody who is interested in the imatinib protocol and also has the bandwidth to participate, we would really appreciate that. But we understand that people are very busy, and if they can't, they can't. Whether or not to wait for another study, I don't have a good idea on when and where those studies would be open, but absolutely that's an option as well.
Great. And then I think I know the answer to this, but is there an option for participating fully remotely? Not quite, huh?
Yeah, that is such a great question. We talk about that a lot. So even a few weeks ago, when we decided that we could get rid of the pharmacokinetics or the drug concentration study on arm 1, we said, okay, let's get rid of that. Unfortunately, the pharmacokinetics are run here, and some of the other studies can only be run here. I am wondering if we get a little further into the study and we get a good idea on the basis of the preliminary results, then yes, maybe we could make changes in the future. but then we will also, lose some information. So for example, the platelet flow assay and the aggregation, bone marrows, those things, at this time, the technology just doesn't exist to have accurate results from those studies unless they're in person.
Okay, great. Someone asked, it's a little different, but can you give Tylenol to a RUNX1 patient for a headache? A little bit different.
So that's a really good question. So usually yes, everybody's different. So I don't want to give individual medical advice on the basis of, people's liver function and others, but normally Tylenol does not interfere with platelet function. Now, this might come, as a surprise to those who have had aggregation done at NIH, because there is a theoretical concern that Tylenol within a certain interval can interfere with that particular study, but that's kind of a research question, and under normal circumstances, for most people, most of the time doesn't affect medical care.
Great, thank you. Someone's asking, let me see if I have any more here. No. Someone is asking, what is the significance of a 1B study? You showed that diagram with the different phases. What is unique? What makes it a 1B specifically?
Basically, that there's already some information available, that it's not a 1A study that hasn't been in people yet, that there's already some data available. It's the simplest way to put it.
And then Dr. Cantor, how did you start sort of this path toward finding imatinib? That's kind of a big question, but I wonder if you could dive back into the history a little bit.
Yeah, sure. There's actually two ways we got to this. One is we were just studying how RUNX1 is normally regulated, and we discovered again that that modification and the particular enzymes that did it were in this class that are inhibited by this imatinib and other drugs like it. And then separately, we did sort of what we call a non-biased screen. We developed a way to just screen a whole bunch of drugs and see if they had increased RUNX activity. And when we did that, I guess it was pretty satisfying. We got quite a few of the top hits were drugs, including imatinib, that all worked in the same pathway and hit the same target. So that gave more confidence that this was the way to do it. And a couple of other groups have also kind of shown some similar findings.
Thanks. I think we're just getting a few thank yous now. People are excited about the study. I guess I'd like to ask Dr. Cunningham, you mentioned that patients could enroll on this did I understand correctly, at their first visit to the NIH Natural History Study as well? I'm wondering about sort of, how will these work together, and is there any concern? How does that affect, the outcomes of the natural history study if they're also taking this medication? Any thoughts there from Dr. Cunningham or others?
Right. So that's an outstanding question. So in terms of being able to start imatinib while, I guess, at their first visit or while they're already here for the natural history protocol. So that would be a little bit tricky because the results take a little bit of time to come back. So the sequencing study usually takes a couple of weeks, for example. And so unless people were already in the area for like 2 weeks or something like that, or they live within driving distance or something like that, then or I guess the other alternative would be if we could get those studies done remotely. So, people theoretically could have an EKG and infectious serologies, with their outside provider. The one thing that we didn't want to do, though, was trigger people's medical insurance and their copays. So, unless there was a clinical reason to do those studies, most insurance wouldn't cover it, and we wouldn't want to put people, in a bind. So all of that to say, those things are theoretically possible depending on the circumstances, but probably 2 trips is better, one for the screening and then one to start the medication. And then you had a second part of the question?
So I mean, I think it's kind of complicated. I'm just wondering if there's any, the other one is a natural history study. So, right. So I'm thinking people are taking all kinds of medications, right? So it's just another, like them being on this protocol is just another factor that is followed. Is that correct? There's no concern about interaction of the two studies or.
Right, yeah.
Mhm.
So that's exactly right. So that the communication between the two studies is important, and that's something that's asked on both studies. What medicines are you taking? So we want to be mindful of that. And then to answer the question of one of the previous participants, is that our expectation is that people go back to their baseline when they stop taking it. And that is something that could also be followed as a specific amendment on the imatinib protocol, or in discussion with the natural history study team